Good morning, my dear students. Welcome to my channel Patel Science Academy. It's our academy by the Shehzad sir. And dear students, in this lecture, I welcome you in the chapter Enhancement of the Food Production as a second number lecture from this chapter. So, dear students, in this lecture, I'm going to explain you about one more technique from this chapter, that is called tissue culture technique. And this technique is very important and very valuable, and having the most uh, important applications in the biotechnology field. So welcome to my academy, Patel Science Academy, and dear students, we'll see all the points in this lecture one by one. So at the end of this lecture, you will be able to know about the tissue culture, its all steps with examples and the applications in a detail. So dear students, let's see it one by one. The Gottlieb Heberlein, this man first pointed out the possibilities of the culture of isolated tissue, that is called plant tissue culture. Actually, by this technique, a small tissue or cell or organ can be isolated from the any part of the plant, or from the any organ of the plant, or any tissue of the plant, and that uh, tissue or that collected or isolated part, if it is cultured in the in vitro condition, then it will be able to produce the whole plant from that small part. So this technique is very interesting technique, and it is called a plant tissue culture. So his original idea, presented in 1902, was called totipotentiality. Theoretically, all the plant cells are able to give rise to a complete plant. So this is very important thing. All the cells present in the plants, they can be they can produce the whole plant. So this technique was pointed out, or it was conceived in 1902 for the first time by this man, that is called Goblet Haberland. So dear students, what is tissue culture? So this is very interesting technique, dear students. Actually, it is a collection of the different techniques. It is in fact emerged as a technique of the plant biotechnology, and here isolated cell or tissue or organs are grown in vitro condition, that is in the laboratory condition, not in the natural condition, on a solid or liquid nutrient medium under the aseptic and the controlled condition of the light, humidity, and the temperature for achieving the different objectives. The part of the plant used in this tissue culture, which is now going to be converted into the whole plant, it is called explant. So all that terms we'll see one by one. First, we'll clear about the tissue culture. What is tissue culture? So dear student, tissue culture means it is the term used for the process of growing the cell artificially means in the laboratory condition, not in the natural body condition. So if you have collected cell from any part of the plant, and if you are going to grow it artificially, means in vitro condition in the laboratory, so this process is called tissue culture. Plant tissue culture, the technique of growing the plant cells, tissues, or organs in an artificially prepared nutrients. That cell you have collected, it has to give the nutrient medium, artificially prepared nutrient medium. It may be solid, it may be liquid, which have the all that nutrients which is required for the growth of that cell, so that that cell can grow and can develop the whole plant. So under the aseptic condition means the condition of the a laboratory in which all facilities are there, and temperature, humidity, and the light condition can be controlled. That condition is called aseptic condition. So simply, the tissue culture technique is the process of growing the cell artificially in the laboratory, and the plant tissue culture is definitely, it is related to the plants. So it is the technique of the growing the plant cells, tissue or organ, and in an artificially prepared nutrient medium in the aseptic condition to produce the whole plant. And the plant cell, it has most important characteristic, that is called totipotent characteristic. What is this totipotent characteristic? We'll see in the next slide. So dear students, this is about the tissue culture technique, and I hope you have understood about that technique. In short, it's about its definition. So origin of this technique is, it was discovered in the early 20th century with the work of this man, that is called Gottlieb Heberland, in 1902. And the technique includes the use of the small cell or tissue or organ of the any plant, so that organ or tissue or cell that has been collected or isolated from that plant can be converted into the whole plant. So simply diagrammatically, this technique is like this. You can see this is that plant. From that plant, this small cell has been isolated, and it has been inserted into this medium, nutrient medium. You can see, we don't need to grow the whole plant by this plant. We don't need this plant, we just need the cell collected from this plant. So with this cell, by this technique, we can produce the whole plants. How we can produce the whole plant? You have to just isolate the cell, and you have to insert into the nutrient medium. When cell will be inserted into this nutrient medium, it will get all the nutrients, sufficient nutrients, and the aseptic condition from that artificial laboratory. And then what will happen? It will grow into the small plantlet, and it will become a complete body of that plant, when and then it can be transferred from this laboratory into the natural field. So simply, this is about this tissue culture technique, and we'll see the detail of this process in the next slides. So dear students, before we go in the depth of this technique, I'd like to explain here some terms which is very famous in this technique, and which is very useful in this technique actually. So the first term it is included in this technique is the explant. Then we'll see about the callus. What is explant? So dear students, explant is an excised piece of the differentiated tissue or organ is regarded as a explant. So dear students, explant means the excised piece, means the piece we have collected from the mother plant, or the we have collected from the plant as a cell, as a tissue, or as a organ. So whatever you have collected from the mother plant, that part which you have collected from the plant for the tissue culture, it is called explant. Suppose for example, the explant may be taken from the any part of the plant. For example, if you have collected the cell of the leaf or the stem or the root, so whatever cell or tissue or organ you have collected either from the root, stem or leaf, so this root, stem or leaf collected cell is called explant. Suppose you have collected the cell from the stem, so that cell is called explant. So I hope you have understood about the explant. Explant means the cell, tissue or organ collected from the mother plant, from any part, maybe leaf, stem and root. Then the next part is callus. And what is callus? Dear students, callus means the unorganized and undifferentiated mass of the plant cell. It is referred as a callus. Generally, when a plant cells are cultured in a suitable medium, they divide to form the callus, that is the mass of the unorganized or parenchymatous tissue. So dear students, callus means, if you have collected explant first, suppose you have collected cell as a explant, so that cell will be inserted into the nutrient medium, and in that nutrient medium that cell will be you will keep that cell in this nutrient medium for few days, about 2 to 3 weeks, and then it will convert into a this cell will convert into a group of cell that is actually the unorganized mass of the plant cell, which is actually not uh, which is not differentiated into the different parts of the plants, means it is not now has differentiated into the root, stem and leaf. So that mass which is undifferentiated or unorganized, there is no organ formation in that mass, so it is just a group of the cell or mass of the cell by the cell division. So that unorganized mass or undifferentiated mass of the plant cell, that is called callus. So in short, explants when divide and redivide, and when it become a group of the cell or the mass of the cell, it is called callus, but it is unorganized, there is no organ formation till then. So it is unorganized and undifferentiated mass. So this is about these two terms, explant and callus. Explant means the part you have collected from the mother plant, that may be leaf, stem or roots, cells, organ or tissue, and callus means the unorganized or undifferentiated mass of the plant cell. So this is about these two terms. Now we'll go for the next slides, and in the next slides we'll see about one more term, that is called cellular totipotency. In the first slide I explained you about that plant cells are known as the totipotent cell. And what is totipotent cell? Now we'll see its definition. So dear students, totipotency is actually it is the ability of the single plant cell to divide and differentiate into the matured plant, means the ability of the plant cell to divide first, then differentiate it into the different parts of the plant to become the matured plant, if placed in the appropriate environment, is called the cellular totipotency. Dear students, it means the cell, if it is having the totipotency, means if it is having the ability to divide and differentiate into the different parts and to become the and it becomes the complete matured plant, if it is given appropriate environment in the laboratory. So that cell which is having this ability, it is called the totipotent cell. So dear students, all plant cells uh, generally has this ability, that is called totipotency ability. So I hope you have understood about this totipotency, means the plant cell has the ability to divide and differentiate into matured plant. So now we'll see about this slide. So in this slides you can see the example of the totipotent cell. As you know, in the body there is a process of fertilization. In the fertilization, dear students, male gamete fuses with the female gamete. So you can see this is female gamete, egg or oocytes, and this is male gamete, sperm. So these are fertilizing, sperm is fertilizing to the egg, and it has become the zygote, and then zygote become the morula. Dear students, zygote become morula. So dear students, this morula you can see here in green color, it is having some cell, and it is having this nucleus, and this is called the morula stage. This morula stage is actually totipotent cell, means now you can understand this morula is having the ability to divide and differentiate into the different parts of the body. So you can see this morula is totipotent, means it can further divide and redivide. So it is dividing here and redividing here and differentiating into the different cells. So you can see here, this is the one layer of the cell, and this few cells are taking its place at one point. So you can see here in the blue color, and this is the layer of the cell. In the yellow color, it is called a trophoblast cell. So this structure in which there is a unorganized mass of this cell here, and one layer, this mass is surrounded by this yellow color layer. So this structure is called blastocyst. So morula has converted into blastocyst because of this totipotency characteristics, because it divides and redivides and differentiate. Now you can see it will it can convert into the human fetus, and human fetus will be in future the human body. And this cell, you can see this cell, the inner mass of this cell, this blue color cell, it is having the pluripotent characteristics. Pluripotent characteristics means it can divide into the different organs too. So you can see here, because it is having pluripotent characteristics, this inner mass is having that characteristic from the blastocyst. So it may be converted into the circulatory system, it may be converted into the nervous system by developing a brain, by developing a heart, and it can be converted into the immunity system to immune system to form the antibodies and antigens. So here, dear students, you can see here this cell is having this characteristic pluripotent characteristics. It means it can differentiate into different organs too. So that's why it is called pluripotent characteristics. And this morula is having totipotent characteristics, means it can convert into this structure too, and it will then become the human being. So this is about the totipotency characteristics, and I hope, dear students, you have understood about this totipotence characteristic. Why I have opened this totipotent and some other terms, explant and the callus, dear students, all that terms are having their use in this technology. That's why I have explained all that terms before I start explaining the different steps of the tissue culture. So now we'll see the different steps of the plant tissue culture. So culturing of the plant tissue, that is again called also called the micropropogation. So this micropropagation or the plant tissue culture, it includes few steps. So we'll see all that steps one by one here, dear students. So step number 1, it includes the selection of the plant tissue, that is the explant, as I told you earlier. So first, definitely you have to collect the material, maybe cell, tissue or organ from the any part of the plant. It may be from the leaf, it may be from the stem, it may be from the root of the plant. So you just collect the plant tissue or cell or organ as a explant from a healthy vigorous mother plant, and this is often the apical bud, but can be other tissue too. So dear students, you are observing here one thing, apical bud. So apical bud can mostly it is used in the tissue culture. Why it is used in the tissue culture, the apical bud, why we use the apical bud as an explant? Because generally it is always uh, disease free part of the plants, because it is not infectious part in the plant. So you can remember it, apical bud is the part which is generally disease free plants, that this is disease free part of the plants. That's why generally apical bud is used for this tissue culture as a explant. So this tissue must be sterilized to remove the microbial contaminants. So this is the first step, and it includes the selection of the explant. And if you have selected the apical bud as an explant, so you just have to sterilize it so that the microbial contamination should be removed before we use that explant. So dear students, from here I will not use the another word, I will just use the explant here, so that it will be easy to understand the ahead portion of this tissue culture. In the next step, dear students, I hope you have understood about the first step. First step is just simply the selection of the plant tissue as an explant and its sterilization. Then the second step includes the establishment of the explant in the culture medium. So you have to establish that selected explant into the culture medium. So dear students, you can see here, this is culture medium, and in the second step, in this culture medium, which is having all the nutrients whatever is required for the development or for the growth of this explant to convert into the callus. So you have to establish that nutrient medium. This medium sustains the plant cells and encourages the cell division. So if you will insert this explant here in this nutrient medium, it will uh, encourage the cell division, because all the enough nutrients are available there in that nutrient medium, so it will be quickly absorbed by the cells and cell will start division. It can be solid and it can be liquid. The nutrient medium can be solid or it can be liquid. Then the each plant species and sometimes the variety within the species, each plant species has sometimes they have the uh, variety, and that a particular medium requirement that must be established by the trial and error method. So this trial and error method means you have to repeat this process maximum time until you get the successful result. So dear students, you can see here uh, uh, you have inserted the explant in this nutrient medium, and if it is not accepting this nutrient medium, and if it is not dividing into the mass, so you have to again repeat this process. So repeat it more and more time until it develops into this mass of the tissue, that is called the callus. So this process is called trial and error method. So I hope you have understood about the second step, in which the explant you have collected in the first step, you have to insert it into this nutrient medium, and that nutrient medium will provide all the nutrients to this explant, and it will start cell division, it will start dividing, and it will then start growing and will be converted into the next step. That next step, you are familiar with that next step, that is called the callus formation. So dear student, now we'll see about the step number third, and third step includes the multiplication of that explant. So dear students, multiplication includes the explant give rise to the callus. As I told you, now explant has converted into the callus, because it has accepted and it has absorbed all the nutrients present in the nutrient medium. The as you know, callus is the mass of the loosely arranged cells, that is unorganized and undifferentiated mass of the plant cell, which is manipulated by the varying sugar concentration and the auxins and the cytokinins hormones. So dear students, this auxin and cytokinins hormone, these are two hormones which we add in the callus. And what happens after adding this auxin in the low concentration and cytokinin hormone in the high concentration, what happens? There is a formation of the multiple shoot and the root system. So the callus may be subdivided a number of the times. So dear students, you can see here this developing callus here, dividing shoot, and in this structure the warmth and the good light are essential for this process. So you can see here, this is the aseptic laboratory, and here you can see the light and the warmth for the dividing shoot and dividing a root system. So now we'll see about the next step. So dear students, I hope you have understood about this third step too. It is for the multiplication of the explant will be converted into now callus, and this callus will be will add the auxin and cytokinin into this callus, and that callus now will go for the organogenesis process, mean it will dividing and redividing, and it will convert into the shoot system and the root system. So we'll see the next step now. Step number 4, it includes the root formation. The shoot are transferred to a growth medium with the relatively high auxin. So dear students, and cytokinin ratio, higher auxin and cytokinin ratio. So students, in the previous slides I have shown you the concentration of the auxin and the concentration of the auxin and cytokinin was like that: auxin low and cytokinin high. When these two hormones are added in this ratio, auxin is low and cytokinin is high, so in that condition shoot system is developed. not root system developed. So here you can see here shoot system has developed but not root system. Why? Because auxin's high concentration is responsible for auxin low concentration is responsible for the shoot system development, and high concentration of the cytokinin is responsible for the shoot system development. So in this step shoot system has developed, and in the next step a root system is going to be developed. Why it is going to be developed? Because the concentration now is relatively changed. What is the concentration now? You can see here the concentration now is likewise the higher auxin and cytokinin ratio. So here auxin is more, that's why the now shoots instead of shoot system root formation takes place and root system is developing here. So this is that root system. So you can see here now, dear students, this root system and shoot system is developing there. The bottle on these racks are young bananas plants and are growing the roots. So you can see here this is the bananas root, developing root by the plant tissue culture. So I hope, dear students, you have understood about this fourth step too. So what happens in this fourth step? The root formation. Why? Because the change of the higher because of the change of the higher concentration of the auxin and cytokinin ratio, and in the previous slides the low concentration of the auxin and the high concentration of the cytokinin was responsible for the shoot system development. So from that callus, root and shoot system now is developed there. So dear students, this is about the tissue culture technique, and now one more time we'll see here the steps in the plant tissue culture or in the callus culture. Dear students, diagrammatically you can see here this is mother plant, means the plant from which you are going to collect this explant. This explant may be the cell, tissue or any organ from the stem, leaf or root. Suppose you have collected the cell of this leaf, so this is that explant. Now you have transferred this explant into this tube. This tube is having the nutrient medium. Now this in this nutrient medium everything is there, whatever is required for this explant for its growth. Now it is dividing, and you can see here now it is going to be converted into this unorganized mass, and you can see this or unorganized mass is called callus. So you can see here this is that unorganized mass which is not differentiated into root system and shoot system. So this simple cell now is converted into this callus which is unorganized mass because of this nutrient medium, because everything is there which is provided in this nutrient medium for this explant to convert into this callus. Now what will happen? The next step includes the formation of the organs. That is why it is called organogenesis. Next step is organogenesis. So what will happen in the next step? Shoot system and root system will develop. Shoot system will develop when there is a low concentration of the auxin and the higher concentration of the cytokinin, while the root system will develop when there will be a higher concentration of the auxin and cytokinin in a ratio. So likewise shoot system and the root system is developed here. Now you can take out this small plantlets from this nutrient medium from that laboratory, outside the laboratory, and you can transport into this pot, and this multiple plants now has been produced, and it is now kept for hardening, and you can transfer it into the plant into the natural field for the natural growth. So dear students, this is the diagrammatic explanation of the tissue culture, and I hope this is very simple and interesting technique, but very interesting and important technique because it has lot of uses, and we'll see all that uses one by one. So the flow chart of this technique, it includes different steps. we'll see now one by one more time. The cleaning of the glasswares, sterilization of the glasswares, and the instruments in an oven or autoclave. This is the first step. Then the second step for the whole tissue culture technique, the selection and the preparation of the nutrient medium, that is ms nutrient medium. Ms nutrient medium means the murashik, and the M for murashik and S for scoop medium. So Murashis and Scoop has prepared this medium. That's why it is called MS medium, with the unknown concentration and the proportion of the different components. So dear students, based on the nature of the explant, there are three types of tissue culture. That is the cell culture, if you have selected cell as an explant; organ culture, if you have selected organ; and the embryo culture, if you have collected or selected or isolated the embryo for the tissue culture. So based on the explant nature, it may be cell culture, organ culture, and the embryo culture. Based on the type of the in vitro growth, there are two types: callus culture in the solid medium and the suspension culture in the liquid medium. So it may be callus culture and it may be suspension culture. If callus culture is there, it means the nutrient medium is solid. If suspension culture is there, it means the nutrient medium is liquid. Maintenance of the aseptic condition, dear students. Aseptic condition is so essential as to avoid the contamination by the other harmful microorganism, and it is accomplished by the sterilization of the glasswares, as I told you earlier. Use of the detergent, hot water oven, nutrient medium by autoclave under the constant pressure of 15 lb per square inch for the continuous 20 minutes, and explant by treatment of the 20% ethyl alcohol and 0.1% HgCl2. Inoculation chamber, the laminar air flow by using the ultraviolet rays tubes for 1 hour before performing the actual inoculation of the explant on the sterilized nutrient medium. So dear students, this is the step by step flow chart of this technique. So you can see here all these steps one by one: the inoculation of the steps, explant in the culture flask containing the sterilized nutrient medium, and inoculation is done in the laminar airflow cabinet unit. Then the incubation of the inoculated explant. Here cells are explant grow, proliferate to form the callus within the 2 to 3 weeks. What happened after the formation of the callus? Subculturing of the callus. If callus is to be maintained to the longer period, callus is divided into the 3 to 4 segments and then transferred to the fresh culture medium. Then it goes for the organogenesis, and I have explained about that: shoot system formation and the root system formation. The initiation of the rooting and shooting that eventually leads to a plantlet means the small plant formation. Thereafter it goes for the hardening. What is hardening? The plantlets are transferred to the polythene bags containing the sterilized soil and kept as a low light and high humid condition. Just remember it: low light and high humid condition for the suitable period of the time, so that it can be converted into the it can be transferred into the natural field. And likewise, the small cell tissue or organ collected from the mother plant can grow into the complete plant by this amazing technique called tissue culture technique. So dear students, this is about this technique. The history of this tissue culture, it includes: in 1885 the Wilhelm Rocks removed the epiblast of the developing chicken embryo and kept it alive for the several days in the warm saline. Though this was the history of this tissue culture technique, and this established the principles of for the tissue culture. You can see here this experiment done by this man was has it had given this the idea for this tissue culture, and the environment that mimicked the natural condition and lacked nutrients, which is why embryo eventually died. So this was the history of the tissue culture. Dear students, you can see here small plantlets producing into these small tubes. So these plantlets can be transferred for the hardening in the in the natural field. Then the applications of the tissue culture. So dear students, as you know, this is the very important and valuable technique of, it is actually the applied technique of the biotechnology and application of the tissue culture. There are various application of the tissue culture in the forestry, agriculture, horticulture, genetic engineering and physiology. The different applications of the tissue culture includes the production of the disease-free plants and the haploid plantlets, micropropagation, production of the secondary metabolites, protoplast culture, the culture of rare plants, zomaclonal variations, and the production of the stress resistance plants. All these steps are considered as applications of this tissue culture, which is having most important value, which is actually having the vital value in the botany. So micropropagation, production of the disease free plants, production of the secondary metabolites, and all other things I told you is considered as applications of the tissue cultures. So steps in the micropropagation. So dear students, this is very important part. Micropropagation or organogenesis or the clonal propagation. Organogenesis via the shoot is considered as one of the most widely used commercial methods of the regeneration of the plants. And what is micropropagation? It is also known as the clonal propagation. It is the only process adapted by the Indian plant biologist in different industries, mainly for the commercial production of the ornamental plants like the orchids, like the eucalyptus, etc., and the fruit plants like the banana, grapes, citrus, etc. So there are different advantages of the micropropagation that we'll see in the next slide. So there are different steps of the micropropagation, dear students, and you can see that steps, the selection and the preparation of the mother plants, sterilization of the plant tissue takes place, the initiation of the culture, explant placed into the growth medium as I explained. Second, multiplication, explant transferred to the shoot media, shoot can be constantly divided. Then the rooting system, the explant transferred to the root media, and then the transfer of the plantlets to the soil, the explant returned to the soil and hardened off. So as you know, as I explained you about that micropropagation step, about this tissue culture. So this is very important part of the tissue culture as a micropropagation. So dear students, this is about micropropagation. You can see these small micropropagules are produced by this micropropagation process. You can see here in this box the micropropagules of the rose plants. It is produced here by the micropropagation, means the callus by adding the auxin and cytokinin can be converted into the root and shoot system, and small plantlet as a micropropagules can be produced by that micropropagation process. So this is about this micropropagation, and you can also see here in this diagram too the excise to the entire shoot system, metameristic region, shoot apex, the cut piece of the tissue from the shoot apex. Now it is, this explant is inserted into this nutrient medium, that it converts into this plantlet as a micropropagules, and it converts into the whole plant. So if you don't want to uh, transport it, this small plantlet can be kept saved in the uh, laboratory as a micropropagules, and then it can be used in the any non-seasonal time. Dear students, you can also see here the micropropagules of this plant. Orchid micropropagation you can see here. So dear students, the micropropagation, it includes different advantages. It helps in the rapid multiplication of the plants. A large number of the plantlets are obtained within a short period and from a small space. Plants are obtained throughout the year under the controlled condition only because of this micropropagation independent of seasons. Genetically similar plants means the clones are produced by this method. Therefore, the desirable characteristics and the desirable sex of the superior variety are kept constant for many generations only because of this micropropagation. It also includes the rare plant and the endangered species are multiplied by this method, and such plants are saved by this micropropagation technique. And it also includes, with the help of the somatic hybridization, that is somatic hybrids, that is hybrid, we are able to obtain the new variety in the very short time span. So this is the magic of this tissue culture, and production of the disease free plants is also possible. It recovers the healthy plants from the diseased plants. Apical meristem as an explant can be used for this. So disease free plant can be produced. You can see banana, potato and sugar cane has been produced by this tissue culture. The secondary metabolites are the organic compound produced by the bacteria, fungi or the plant which are not directly involved in the normal growth, development or the reproduction of the organism. So secondary metabolites, dear students, are the only organic compounds produced by the bacteria, fungi or the plants which are not directly involved in the normal growth. So they are the organic compound produced by these microorganisms or plant. Catharanthus roseus plants you can see as a wind blasting and wind cristines as a secondary metabolites of this Catharanthus roseus plants. Then the Daucus carota plants, anthocyanin as a secondary metabolites of these plants you can see here. Then the stramonium plants, the tropane alkaloids. These tropane alkaloids, actually these are the ester alkaloids resulted from the coupling of the organic acids with the amino alcohol base. The parent base is the tropane base. So this is the secondary metabolites of these plants. Then the mentha piperata plants, so you can see as a secondary metabolites of this plant is also possible to produce the possible only because of the tissue culture. Nicotiana tabacum plants, this product has been produced as a secondary metabolites by this tissue culture. Somatic hybridization technique we'll see in the next lecture, dear students. So this is about this tissue culture technique, and in this lecture I have explained you about the tissue culture techniques, is its different steps and its magical uses and the examples of the plants that is produced by the tissue culture technique. Dear students, the high yielding varieties of the banana, that is Shrimati, Basarai and the G9, are mostly used in Maharashtra in India by the tissue culture technique. So dear students, now at last, if you summarize the topic, the tissue culture actually it's a technique of growing the new plant from the any part of the mother plant as a cell. It may be produced from the cell, from the tissue, from the organ collected from the any part of the plants, from the maybe from the root, stem and the uh, leaf. So this is tissue culture, and it includes four main steps. In the first step you have to collect or you have to isolate the explant, and then in the second step you have to transfer it into the nutrient medium for the culturing process. Then it converts, then it divides and redivides and converts into the callus. Then it goes for the third step, that is called organogenesis. And in the organogenesis, the process micropropagation takes place, and that micropropagation, because of that, adding the auxins and cytokinin hormone in the different concentration, in the different ratio. If low auxin is there, high cytokinin is there, there will be a formation of the shoot system. And if high auxin will be there, there will be a formation of the root system. So likewise, the small propagules, small plantlet is produced, and then it is transferred in from that laboratory into the uh, pots, for hardening process. And it can be, then that small plantlet can be transferred into the natural field for the natural growth. So dear students, this is about this tissue culture technique. So thank you very much for watching this video. I would like to do one announcement here. Because of your support, my channel Patel Science Academy has crossed more than 1500 subscribers. So thank you very much for that. And dear students, keep spreading my link with your all friends. And please do subscribe my channel, like my videos, share it with your friends and with all the people who want to learn about that topic from the biology. And like it, share it, and comment for your queries. And if you want to have some new video for some special topics, so you can comment me. I will try to upload that video for that particular topic too. And please press the bell icon so that you can get the instant notification of my new video. So thanks for watching. Thanks a lot.